all the bits back together. Okay. So the trick here is not to press the white button.
Press the white button.
Okay. Thank you. So good morning. Just by way of introduction, I'm Stuart Legrice. I work for at the National Cancer Institute in Frederick. I started my retrovirology career about 25 years ago at Hoffmann-La-Roche in Switzerland. My arrival coincided with the arrival of HIV. There was no other coincidence beyond that. But I spent 7 years at Hoffmann-La-Roche working on development of antivirals and diagnostics for HIV. I moved from there to Case Western Reserve University in Cleveland in 1990. And I spent 10 years at Case. I was director of the Center for AIDS Research. Again, 10 years dedicated to multiple aspects of HIV infection. And in 1999, I got a telephone call. I always say, when you've lived in Cleveland for 10 years and you give a lecture, it's nice to be anywhere else in the country in January when you give a lecture except Cleveland. No offense to the Clevelanders. But in 1999, I was asked to join the National Cancer Institute, and I'm currently the head of the Center of Excellence in HIV, AIDS, and Cancer Virology. So with that introduction, you'll probably gather I'm not from these parts, and I hope you've got used to my accent. I was asked to give a summary, a recap of the recent workshop that we held here in Washington. I actually, a year ago, organized the first XMRV workshop at the NIH. And this was in response to a few papers at the Cold Spring Harbor meeting on retroviruses. XMRV was not a major problem then. The issue that we were dealing with was the percentages in the control population. About 4% of the control population was testing positive for XMRV. And simple math said that could represent about 10 million Americans. And we felt that at the time it was worth putting a group together to understand the implications of this and provide guidelines and recommendations for the leadership at both the NCI and the NIH. So we moved on to the third meeting. There was one in between in November in Bethesda, and this year we had the first international workshop on XMRV, and that's what I'm going to report on today. I wanted to say a couple of things at the beginning. About 2 or 3 days before the meeting, I read in the press that as a scientist, I and others were in a frenzy about XMRV. I sometimes get very tired working 7 days a week on XMRV. I get sometimes frustrated when my colleagues are not around when I need them, but there is certainly not a frenzy with XMRV. I think we're going to see today that there are disparate results, and as scientists, we're trying to understand where methods that look as accurate with one researcher as the other turn out to find XMRV and not find XMRV. So there is no frenzy amongst us. Another point that came out of the meeting, it got rather contentious, the meeting. But I think that the statement made by Jonathan Stoy from the United Kingdom is very important. That as scientists, we are under no mandate not to find XMRV. We are under no mandate not to find an association of XMRV with either chronic fatigue syndrome or prostate cancer. We're under a mandate, as I said earlier, to understand why there are such disparate results in the literature. The third point I wanted to say is the talks I'll be summarizing today are those that, for which we were given permission to present, to publish on the web. A number of talks I'll not be summarizing today because those speakers, for a variety of reasons, they're going to be published soon, declined to allow us to put them on the web. So this was the basis of the meeting then. Let me see. There were 6 topics. I'm going to give you, let me show you my next slide. Maybe this will help explain where I'm going here. This just shows the rise in the number of papers, the XMRV publications, over the last 4 years. Okay, we had the 2006 paper from the Cleveland Clinic Group associating XMRV with prostate cancer. I could actually finish my talk here and leave you to work out the problems that we are dealing with, because this slide shows as many reports finding XMRV as reports that don't find XMRV. At the time of the meeting, John Coffin, I think, said there was about 8 reports could find it, 8 reports couldn't find it. We're also trying to understand disparities, like one report finds it in a large percentage of patients, another report finds 0 out of 800. These are at either end of the spectrum, and so clearly there's something we don't understand about XMRV. But as I said, as of September, there's probably been 2 or 3 added on to this. There's about 45 to 50 publications, and we are split rather evenly between groups who can detect it and groups who can't detect it. So I'm going to have to give you an introduction on gamma retrovirus biology, and I hope you'll follow the thread here, because the issues of potential contamination and sample handling, reagent contamination, are real issues that I think for the last year we've not really wanted to talk about, and I think this was one of the first times where we got a chance to sit in an international audience and really discuss these issues, because they are very serious in terms of detecting XMRV. But to do that, I have to give you a little lecture on gamma retroviruses. This was given by John Coffin from Tufts University. Oh, does the red take me back, or does it do? Oh, yeah. So gamma retroviruses are very simple viruses. XMRV is a gamma retrovirus. They're very simple retroviruses. They have only GAG pollen envelope genes. They're very widespread in nature, and isolates from birds, mammals, and reptiles have been identified. They readily give rise to endogenous proviruses. They're most often transmitted vertically from the mother to the offspring. They cause a wide variety of diseases: cancer, neurological, degenerative, and immunodeficiency diseases. Cancer is normally a result of insertional activation of proto-oncogenes. And this is a very important point. You'll see the next 2 or 3 slides will have one red point made amongst a number of black, and I want to try and string those together. Gamma retrovirus infection is lifelong, and unlike HIV, there are very few replication cycles of a gamma retrovirus in the host. And this is actually, if we do get to the point of antiretroviral therapy, this is a very important point for XMRV. Xenotropic murine leukemia virus, XMLV. XMLV is inherited as an endogenous provirus in all inbred mice. Many more proviruses have been found in mus musculus species, in the wild mus musculus species. Some proviruses are intact and infectious. Xenotropic MLV can infect almost all mammals except some species of mus due to the receptor polymorphism. They're not directly pathogenic in mice due to the lack of the receptor, but their long-terminal repeat, this is a hallmark of a retrovirus. It's often found in oncogenic recombinant MLVs. Now here's one of the first points that I think is very important. XMLV is a common contaminant of human tumor cells due to passage through nude mice. This is something that for decades as scientists, immunologists have been using. We're talking about XMRV, which is related to murine leukemia virus. We're now talking about, for maybe 2 to 3 decades, labs using nude mice that contain endogenous proviruses. And related MLVs cause a variety of diseases, malignant immunodeficiency, and neurological in mice. So I think you'll see where I'm going here. I'm now getting to polytropic and modified polytropic murine leukemia viruses. This was the subject of the recent paper by Lowe and Alter in PNAS. Modified and polytropic MLV are endogenous proviruses. They're in all inbred mice. They're found in wild mus musculus subspecies. This is very important. No infectious polytropic MLV has been discovered to date. And again, I'm going to thread this theme through a couple of my slides relative to the Lowe and Alter paper that have discovered polytropic MLV sequences. So far, none of the polytropic MLVs have been shown to be infectious. They can infect virtually all mammals, using both forms of the XPR1 receptor. They're not directly pathogenic in mice, but the envelope gene is often found in oncogenic recombinant MLVs. The pathogenicity in other species is unknown. Both types derived independently from xenotropic murine leukemia virus in response to the mutation of the receptor, XPR1. And they suffered a number of significant mutations, and that's mediated by the mouse ApoBec3 protein. That's an innate host protein that is used for defense against HIV, and the mouse ApoBec protein is actually very effective against polytropic and modified polytropic murine leukemia viruses.